Hello, I'm Professor Anthony Isles from the Division of Psychological Medicine and Clinical Neurosciences in the School of Medicine at Cardiff University, and today I'm going to be giving a lecture on genomic imprinting in human disease. So at the end of this lecture, I hope we'll have a good understanding of what genomic imprinting is, and I'm going to give some definitions and historical background to the idea of genomic imprinting and talk about the imprinting mark, how it is erased during gametogenesis, and how it's maintained during somatic cell division. And then we'll move on to examples of disorders linked to imprinted genes. And today I'm going to talk about Beckwith-Biedermann syndrome and Silver-Russell syndrome, Angelman and Prader-Willi syndromes, and how duplications at chromosome 15q can give rise to psychotic illness depending on their parent of origin. And by understanding these disorders associated with imprinted genes, we will also have an indication of the implications of genomic imprinting for genetic counselling. So we'll be talking about de novo versus inherited mutations, and how, if the mutations are inherited, they may skip generations, and how we can use epigenetic-based screening in order to determine the parent of origin of any specific mutation. And then finally, we'll finish with a brief discussion of how the incidence of genomic imprinting related disorders may be increased following assisted reproductive technologies. So in the early 1980s, two developmental biologists were interested in the question of whether the parental genomes were equivalent. So Azim Surani, based in Cambridge, and Davor Salter, based in Pennsylvania, used mouse models to investigate this question. And this is an interesting question and quite important because it's effectively Mendel's second law of inheritance, so Mendel's law of equivalence, which states that no matter which parent contributes a gene to its offspring, the gene will behave in the same way. And at this time, we understood that for mammalian development to progress, it was necessary for an individual to have two copies of the genome. So haploid individuals would not develop, individuals with one copy of the genome. So we knew that it had to be two copies of the genome, but the question they were asking was whether it had to be two copies of a maternal, or two copies of a paternal, or a copy of each. And so they set up these experiments where they could take advantage of recent developments in nuclear transfer technology and things like this in the early 80s to generate parthenogenetic, gynogenetic embryos, which had two copies of a maternal genome, or androgenetic embryos, which had two copies of a paternal genome. And so they did these experiments and followed the trajectory of development of these mouse embryos, and what they found was that they were not viable. These embryos failed to reach mid-gestation in the mouse and had quite striking morphological differences. So in the parthenogenetic embryos, there was an underdevelopment of extra embryonic tissue, so the placental tissue. In the androgenetic embryos, the embryos were really severely underdeveloped and had exaggerated development of these extra embryonic tissues. So the answer to their question was, of whether the parental genomes were equivalent, was no. And so they then proposed the idea of a mark on these different parental genomes. So specifically, quoting the Surani paper from 1984, we propose that specific imprinting of the genome occurs during gametogenesis so that the presence of both a male and a female pronucleus is essential in an egg for full-term development. And so this is where we get the term genomic imprinting. It refers to some kind of marking of the genome which gives rise to differential recognition of the maternal and the paternal genomes. And this was not just limited to the experimental mouse models that they used in these pioneering experiments. Later studies also investigated two different types of cancer that can occur. The first one was the benign ovarian teratoma, and this is a rare germ cell tumour, and it consists of different tissues derived from all the different germ layers, so you can have hair and skin and teeth in these different tumour types. And most of these ovarian teratomas are benign, but the key thing was that most of these teratomas also have two maternal genomes. So these were effectively equivalent to the parthenogenetic embryos that were developed in the lab. Similarly, a hyatidiform mole is a rare gestational trophoblastic disorder. It's basically a disorganized mass of placental tissue without a fetus. But again, most of these are benign. But importantly, most of these have two paternal genomes. So these are equivalent to the androgenetic embryos that were developed in those experiments. And so we now know that the reason for this failure to develop is the presence of what are termed imprinted genes. So like all other genes in the mammalian genome, they are inherited from both mum and dad. So a copy is present from mum and dad. But these imprinted genes are differentially marked in order to distinguish the parental genomes. And the consequence of this differential marking is that individual imprinted genes are expressed from one parental allele only. So for some imprinted genes, the example here being UB3A and CDKN1C, it is only ever the maternal copy that is expressed, and the paternal copy that is inherited is simply silenced. However, for other imprinted genes, nectin, PEG3 being examples, the opposite is true. So here, only ever the paternal copy is expressed, and the maternal copy that is inherited is silenced. So we know now that there are about 260 imprinted genes known in the mouse, and about 200 or so, just slightly shy of that number, known in the human. And there is some homology, some conservation, but it's not complete. And these imprinted genes are distributed across the genome in humans and in mice. They're not present on any particular chromosome, so they're spread across the different chromosomes. In the mouse, it seems that roughly 50% are maternally expressed and 50% are paternally expressed. In the human, there is this apparent bias towards paternally expressed genes, but this may actually be an ascertainment bias because it's very difficult to determine the imprinting status of a gene in human tissue, unlike it is in the experimental model. So this may actually skew, well, as we discover more imprinted genes in human, this may even up to be more like 50-50. There's no reason to think that there would be an increased number of paternally expressed imprinted genes in the human. So what is this imprinting mark? So just to go back to the definition that was defined by Serrani, we propose that there's a specific imprinting of the genome occurs during gametogenesis, so that the presence of both a male and a female paranucleus is essential in an egg for full-term development. So the nature of this imprinting mark is epigenetic. So what do I mean in terms of epigenetics? So the original definition that we use was developed or was derived from this quotation from Conrad Waddington in about 1942. The transmission and perpetuation of information through cell division that is not based on the sequence of the DNA. And so Conrad Waddington was interested in how different cell types that contain the same DNA become specialized into different tissues. And he proposed that there was some additional layer of information that gave rise to, that provided information with regards to which genes are expressed in which cell. And so that's the idea of epigenetics broadly. And for our understanding of imprinted genes, the key epigenetic mark that we know about is the addition of methyl groups to the cytosine nucleotide base of DNA. So cytosine can be in several different states. This is the normal cytosine molecule. This is the methylated one. You can also have something called a hydroxymethylation, which is a kind of intermediate between methylated and unmethylated. And in the case of imprinted genes, these cytosine methyl bases occur in runs of cytosines, which are termed CpG islands, and the specific term for the methylated region that occurs in conjunction with imprinted genes is DMRs, differentially methylated regions. And these are the key epigenetic mark that distinguish the parental genomes. In addition to DNA methylation, you may see in the literature, and I may refer to later on, the idea of the packaging of the DNA also being an epigenetic mark. So DNA is not just loose in the cell, it's tightly compact in order to protect it. So the DNA is wrapped around what are known as nucleosomes, and the nucleosomes themselves are packed tightly to form chromatin. The chromatin then is folded and condensed further to form the chromosomes. And depending on how the 3D structure, the 3D packaging of the DNA, depending on how open that is, gives rise to whether that gene is more likely to be transcribed or to be silenced. But for the purposes of this lecture, we're going to mainly be talking about DNA methylation that occurs at these DMRs, these differentially methylated regions. And they're the key component of the imprinting mark. So here is a schematic of an imprinted cluster. This is the imprinted cluster that occurs on human chromosome 11p15. And it's a good representation of a large number of imprinted clusters spread throughout the genome. So what you can see is that there are both genes that are expressed only from the maternal copy, so these are the ones that are in red. And at the same locus, there are genes that are only ever expressed from the paternal copy, and these are in blue. And then either side of the imprinting locus, you have biallelic expressed genes, which represent the genes that are seen in the vast majority of the genome. And so, of course, these maternally expressed genes, the copy on the paternal chromosome of these genes is silenced, and that is represented by the white boxes. In addition to the genes themselves, we can also see the presence of differentially methylated regions, DMRs. And so at this locus we actually have two. The first is the H19 DMR, and this DMR is methylated, heavily methylated on the paternal chromosome, but unmethylated on the maternal chromosome. And this particular DMR appears to control the expression, the imprinted expression of H19 and IGF2. H19 being a non-coding RNA that's paternally expressed and paternally silenced, IGF2 being a protein coding gene that is paternally expressed and maternally silenced. This imprinting locus also has a second DMR, which is called the KV DMR, and this is heavily methylated on the maternal chromosome and unmethylated on the paternal chromosome. And this DMR is particularly important for the control of the expression of CDKN1C and KCNQ1. And so one take-home message from this is that the DNA methylation that occurs at the DMRs is not necessarily directly related to the gene expression. It is simply a mark that distinguishes the maternal and paternal genome. And there are other epigenetic processes that build upon this mark and give rise to the changes in gene expression. So an important consideration here is that genomic imprinting is a developmentally determined process, and so it goes through a certain cycle during embryogenesis. So at the beginning here we've got a schematic showing a DMR at this genus XL. And this DMR is maternally methylated, so here we're seeing a single copy in an egg that's heavily methylated, and the DMR on the paternal genome is unmethylated in this particular locus. So here we can see a sperm where this DMR is unmethylated. Following fertilization, this inherited methylation status on the maternal chromosome is maintained. And built upon throughout somatic cell division. And so as embryogenesis progresses, the methylation is maintained and built upon with other epigenetic marks, and then in cells in which genus XL is expressed, it is only ever expressed from the paternal copy. To look at that in a little bit more detail, we just need to get an understanding of the process of DNA methylation change during the course of embryogenesis. And this schematic shows the level of methylation that occurs at the maternal and paternal genomes throughout the course of gametogenesis and also following fertilization during early development. If we just focus on this time point onwards here, we can see that the paternal genome, which is found in the sperm, is heavily methylated. So the whole genome is methylated at about 90% levels, whereas the genome of the egg, the female, the maternal genome, is methylated at around 40%. And what happens generally following fertilization is that there is a rapid global demethylation of the paternal genome and a slightly slower demethylation of the maternal genome. And then as development progresses further to the blastocyst stage, then methylation is reset on these across the genome according to cell-specific status. So the tissue-specific and stage-specific methylation is set up across the genome. So that's the status for the whole genome. But what happens for those few imprinted genes, those 200 or so imprinted genes, and their associated DMRs, is that they are protected from this demethylation. And their level of methylation stays at 50%. So fully methylated on one genome, one parental genome, and unmethylated on the other parental genome. And the methylated DMRs are protected by a whole group of proteins, the SCMC proteins, and also this molecule here, ZFP57, which literally sit on the DMR and protect it from this global demethylation process. And this is important, we'll come back to this at the end. But the point is that the status of imprinted genes, the epigenetic status of imprinted genes, is distinct from that of the rest of the genome, in that it's inherited from mum and dad and then maintained throughout somatic cell division. However, in addition to the development of the somatic cells during embryogenesis, you also have gamete development. And here the situation is slightly different and depends on the sex of the developing embryo. So if we take a female embryo, what happens is that inherited imprinting mark is stripped away during oogenesis and then reset as is required for the sex of that developing embryo. So if that embryo is a female, it's going to produce eggs and a maternal genome, and so therefore it needs to be heavily methylated. However, if that embryo is a male, what happens during gametogenesis is that inherited methylation mark is stripped away and then reset according to the sex of that developing embryo. And this embryo is going to be a male, it's going to be a father, produce sperm, and so the DMR here for this particular locus is unmethylated. And then this gives rise to the next generation. And this process of resetting the imprinting mark during gametogenesis is important when we consider the impact of inherited mutations that give rise to disease and how they may skip generations. And we'll come back to that at the end. So obviously, given the status of these imprinted genes, we can see that if there are mutations which affect just one copy, the active copy of a given imprinted gene, it's highly likely to give rise to disease. Because what you could see is, for instance, if a mutation were to affect UB3A, and it was the maternal copy of UB3A, and this mutation either prevented expression or rapidly increased expression, then this would be causal of disorders and mutations and other associated phenotypes. And so we can see that there are lots of these imprinted genes that are scattered through the genome. And although they're very small in number, there's only 200 odd genes, mutations that affect them can be quite damaging. So many of these imprinted loci are associated with different diseases, different mutations, and these mutations can be genetic or epigenetic in nature. And we'll talk about this a little bit more. Critically, and possibly because they are so damaging and penetrant, imprinting disorders are rare and are often de novo, so they often occur spontaneously. But they can be inherited, as we will see. And so there are broadly 4 types of genomic and imprinting defects that can give rise to imprinting disorders. So in this schematic here, this is the normal status of this imprinted gene. And what we have is a gene that is expressed from the paternally inherited copy. And there is a DMR associated with that gene, and is represented by these lollipops. The DMR is methylated on the maternal chromosome, but unmethylated on the paternal chromosome. And that is the imprint that distinguishes the paternal and maternal allele for this particular gene. So the first mutation that can give rise to a change in the expression of this particular imprinted gene here, this hypothetical imprinted gene, is a uniparental disomy, a UPD. And this is where, during the course of very early embryogenesis and cell division, there ends up being 2 copies of one parentally derived chromosome. So what can often happen is that DNA replication is not quite complete or is inappropriately conducted, and so a cell ends up with 3 copies of a given chromosome, 2 derived, in this case, from the mother and 1 derived from the father. And in that case, what sometimes happens is that these cells then kick out the rogue chromosome, and if they kick out the paternal chromosome, then you end up, in this scenario, with 2 copies of a maternal chromosome at this given chromosome. So that means that the methylation status associated with that chromosome is also inherited, and consequently the expression of that gene is absent because of the methylation status associated with maternal UPD in this particular case. And so there is no expression of that gene, it is completely lost. And although there are two functional copies, because of this epigenetic status, this imprinting mark, those copies are simply not expressed. The second mutation type, which can give rise to imprinting disorders, is basically a copy number variation, a CNV. And this could be a deletion, as occurs in this example here, or indeed a cis duplication of an imprinted gene. So in this particular case, what we've got is a CNV deletion that has taken out this gene, but only on the paternal chromosome. And again, although there is a functional copy on the maternal chromosome, this functional copy is silenced because of its imprinting status. And so this CNV deletion means that the product of this gene is completely absent. And that can obviously be quite damaging. The final genomic mutation that can give rise to an imprinting disorder is an SNV, a single nucleotide variation. So damaging SNVs, which give rise to changes in protein structure, so amino acid content and protein structure, are the general mutations that can occur throughout the genome. But if they hit an imprinted gene and are affecting the normally expressed copy, then that is highly damaging because, again, although there is a functional copy on the maternal chromosome, its epigenetic status means that it's silenced, and so the only copy that is expressed is this damaged version of the protein. And so this can be highly penetrant when it occurs at imprinting loci. Finally, imprinting disorders can also occur as a consequence of what are termed imprinting defects, or more broadly, epigenetic mutations, epimutations. So here what you have is the methylation status that is associated with this loci, for some reason, is abnormal, incorrect. And so in this case, what we see is hypermethylation of the normally unmethylated paternal DMR. And so although the DNA remains completely unchanged, these are both functional copies of this gene. Because this methylation mutation has occurred, this hypermethylation here, this means that this gene is not expressed, and so the product of that gene is completely absent, and this can be quite damaging. So, as I said, there are only about 200 imprinted genes in the human genome, but these can be quite damaging, any mutations that occur with them, and there are a large number of disorders that are associated with these imprinted genes, and these are broadly listed here, and they seem to hit certain physiologies. So in particular, they seem to be impacting on placental physiology and fetal growth. And then postnatally, we also get effects on growth, endocrinology, muscle structure, brain development, the instincts for feeding and nutrition, which could be related to changes in endocrinology and also changes in the brain. And also there are some skeletal changes as well. So I'm going to give some examples of a couple of imprinting disorders to illustrate both the types of phenotypes that are associated with these imprinting disorders broadly, but also to illustrate how the different mutations may give rise to these disorders. And this will then inform a brief discussion of how these might be important for have important considerations for genetic counselling. So the first two disorders that I'm going to talk about are Beckwith-Wiedemann syndrome and Silver-Russell syndrome. So these two disorders are associated with the imprinting cluster on chromosome 11p15 that we saw previously. And again, this imprinting cluster has several genes that are maternally expressed only, and then a number of genes that are only paternally expressed. And it's also associated with two different DMRs. The KV DMR that's heavily methylated on the maternal chromosome, and the H19 DMR that's heavily methylated on the paternal chromosome. And these are both important for the regulation of expression. So Beckwith-Wiedemann syndrome and Silver-Russell syndrome are both broadly growth disorders. So Beckwith-Wiedemann syndrome is rare, occurs in about 1 in 14,000 live births, and the babies are generally born with overgrowth. They're in the 95th weight percentile at birth. They also have a number of different phenotypes associated, such as macroglossio, a large protruding tongue. They have a number of abdominal wall defects and predisposition to Wilms tumour. They also have a large placenta, and they have a number of endocrine problems related to insulin signaling. Silver-Russell syndrome, in contrast, these individuals are born very low, so these are born below the 5th percentile. Silver-Russell syndrome is extremely rare. It occurs in 1 in 100,000 live births. And as well as being born with low birth weight, these individuals fail to thrive in terms of their postnatal growth. They also have a number of morphological phenotypes. They lack subcutaneous fat, and they also suffer from night sweats. So to understand how these imprinting loci give rise to these two growth disorders, we need to understand a little bit about what the genes in this locus encode. So the maternally expressed CDKN1C is a cell cycle inhibitor which inhibits cell division and growth. So expression of this gene is important for reducing growth effectively. In contrast, IGF-2, which is the insulin-like growth factor, is a growth factor which promotes cell division and particularly fetal growth. And so these two genes are almost having an antagonistic effect on growth. So what kind of mutations lead to Silver-Russell syndrome or Beckwith-Wiedemann syndrome? So the first mutation I'm going to consider in relation to Silver-Russell syndrome, and this is the undergrowth syndrome that we saw, is maternal uniparental disomy, so UPD. So this is where the individual has two maternally derived copies of chromosome 11. And of course, the consequence of that is that they have both chromosomes have the epigenetic status for a maternal chromosome, so heavily methylated at the KV DMR, unmethylated at the H19 DMR. And this means that IGF-2, which is normally paternally expressed, is completely absent. And CDKN1C, which is only maternally expressed, is overexpressed. There are two copies expressed here. Beckwith-Wiedemann syndrome, which is the overgrowth syndrome, can also occur as a consequence of uniparental disomy, but in this case, it's paternal uniparental disomy. So two paternally derived copies of chromosome 11. So here you have the opposite scenario, in that you have two active copies of IGF-2 as a consequence of the methylation status of the H19 DMR. And so you get 2 times the level of IGF-2. But because of the absence of methylation at the KV DMR, you get no CDKN1C expression. at all. In addition to those UPD mutations, which are the most prevalent mutations that give rise to Silver-Russell syndrome and Beckwith-Wiedemann syndrome, there are a number of other mutations that can occur, and I'm going to detail these in relation to Beckwith-Wiedemann syndrome. So the first of these is a point mutation, an SNV, which hits CDKN1C and causes it to be non-functional. So here again, although you have a functional copy of CDKN1C, it's silenced because of its methylation status here at the KVDMR. And the active copy of CDKN1C carries this mutation, which means that it's non-functional. You can also get effectively CMVs which hit this, and if they're maternally derived, they're going to give rise to loss of CDKN1C expression. And you can also have methylation mutations, particularly methylation at the KVDMR, but you can also loss of methylation at the KVDMR, but you can also have hypermethylation at the H19 DMR. And so if we take the example of the loss of methylation at the KVDMR, here, although effectively the chromosome's complement is correct, you have a maternally derived and a paternally derived chromosome, for some reason the methylation that occurs on the maternal KVDMR has been lost, and the consequence of that is that its epigenetic status basically looks the same as a paternally derived chromosome, and the consequence of that is that you get no expression of CDKN1C, and the expression profile is similar to a paternal chromosome at this region. It's important to note that the DNA sequence here is not mutated, so you have functional copies of CDKN1C, but it's the associated loss of expression of CDKN1C that gives rise to the disorder. And so you can see this here. These are identical twins, so genetically indistinguishable, but one of the twins here on the right-hand side has Beckwith-Wiedemann syndrome, and that's because of an epigenetic mutation that gives rise to changes in the levels of expression of CDKN1C. So basically, we can boil this down to the fact that Beckwith-Wiedemann syndrome is caused by too much IGF-2 and or too little CDKN1C, so too much of this insulin-like growth factor and too little of this cell cycle inhibitor, CDKN1C. In contrast, Silver-Russell syndrome occurs as a consequence of too little IGF-2 and or too much CDKN1C. So too little of this growth factor and too much of the cell cycle inhibitor, the growth inhibitor. So that's Beckwith-Wiedemann and Silver-Russell syndromes. We're next going to talk about two other syndromes that occur at a different mutations at a different imprinting locus, and this is the imprinting locus at chromosome 15, Q11 to Q13. And these syndromes are Prader-Willi syndrome and Angelman syndrome. And then, in addition, I'm also going to talk about duplications at this region and give psychotic illness. But for the first part, we're just going to focus on these two different disorders. So here is the imprinting locus at chromosome 15q11 to q13. Again, it contains a number of imprinted genes, and again, it contains genes that are only ever expressed from the paternal copy, here showed in the blue boxes, and other genes that are only ever expressed from the maternal copy, so again shown here in the red boxes. The paternal copy of this gene is silenced, and the maternal copy of these genes is also silenced. And then either side, you have a number of biallelic expressed genes like normal. there is a DMR that is associated with this locus, and this is called the Prader-Willi IC, imprinting center, confusingly, but it is basically a DMR. And in this case, the DMR is heavily methylated on the maternally derived chromosome, as represented by these black lollipops, and unmethylated on the maternal chromosome. So Angelman syndrome is a neurodevelopmental disorder characterized by quite severe delayed development, little or no speech, motor problems, and frequent seizures. Prader-Willi syndrome is also a neurodevelopmental disorder. There is some developmental delay, but it's not as severe as was seen in Angelman syndrome. But really, Prader-Willi syndrome is characterized by a hypothalamic dysfunction. So this is a region of the brain that controls feeding, and these individuals have an insatiable appetite that is very difficult to manage. They also have a number of behavioural problems, obsessive compulsive disorder, and some, as I said, quite mild learning difficulties. So this is the Prader-Willi Angelman syndrome locus again. And what is important to realise is because this region contains a number of repeat elements, it seems to be quite unstable. So you have these characteristic breakpoints, breakpoint 1, 2, and 3. And what can happen here is that deletions can occur between these breakpoints. So either between breakpoint 1 and 3, or between breakpoint 2 and 3. And of course, any deletions that do occur at these chromosome interval will affect these imprinted loci. So if a deletion CMV occurs on the paternally derived chromosome, so expression of these paternal genes is absent, then it will give rise to Prader-Willi syndrome. In contrast, if this deletion CMV occurs on the maternally derived chromosome, and so impacting the expression of these normally maternally expressed genes, then it gives rise to Angelman syndrome. And it's because of the presence of these imprinted genes at this interval that this same mutation, these same deletion CMVs can give rise to quite very distinct disorders. In addition to the primary cause of these two disorders, which is a deletion CMV occurring at this interval, if we do a deep dive into Angelman syndrome, I can outline a number of other mutations which also give rise to Angelman syndrome as a disorder. So maternal deletion CMVs occur in about 75% of the cases. But as I said, in addition, you can get a number of other mutations which give rise to this disorder. The first is a damaging SMV that affects the maternal copy of this gene UB3A. So UB3A is clearly the critical gene for Angelman syndrome, and a damaging SMV that affects this gene that is on the maternal copy will give rise to Angelman syndrome. So even though there is a fully functional copy of UB3A on the paternal chromosome, because it is silenced because of the nature of the imprint, all functional copies of UB3A are absent. In addition, but quite rare at this interval, you can also have a uniparental disomy. And in this case, if the uniparental disomy is paternally derived, because of the imprinting status that's on this chromosome, so the absence of methylation here, the expression on the chromosome is of the paternal nature, and so you lose any expression of the maternal UB3A. Finally, you can also get an epimutation, an imprinting defect, and this is where both paternal and maternal chromosomes are present and the genes within them are correct, but the epigenetic status at this 15q11 to q13 interval and at this DMR in particular is abnormal for some reason. And in this particular case, if it's hypomethylation, so an absence of DNA methylation on the maternal chromosome, that then leads to these genes being expressed as if they were the paternal chromosome, and again leads to an absence of expression of UB3A. And so we can basically say that these two disorders are caused by different effects on the maternal and paternal genome at this locus. Angelman syndrome is caused by de novo loss of maternal gene expression due to either epigenetic or genetic mutations, whereas in contrast Prader-Willi syndrome is caused by de novo loss of paternal gene expression due to epigenetic and genetic mutations affecting this imprinted gene cluster. So, in addition to deletion CMVs, which can occur at this interval between these breakpoints, very rarely you can also get duplication CMVs. So this is where the region between breakpoint 1 and breakpoint 3 is effectively duplicated in cis, or the region between breakpoint 2 and breakpoint 3 is duplicated in cis. And the consequence of that can also be damaging. So in 2011, we published a study looking at the incidence of duplications at this interval in individuals who have schizophrenia or a psychotic illness that's like schizophrenia, schizoaffective disorder. And what we found was that out of about 3,000 individuals, there were about 6 or 7 of those individuals with schizophrenia who had what was a maternally derived duplication at this interval. So a duplication of this region here gave rise to psychotic illness. What we didn't know, though, because we didn't find any, was whether the paternal duplication was damaging too, or whether it's simply the case that any duplication at this interval would have given rise to psychotic illness, or whether it's only the maternal duplications. So then we did a larger study, again looking for maternally and paternally derived duplications, and what we found was that maternally derived duplication CMVs were associated with schizophrenia, so an increased incidence. If you had a maternally derived duplication, you had a far increased incidence of schizophrenia and a likelihood to develop schizophrenia. And we did find some paternal duplication CMVs, but these were not associated with an increased incidence of schizophrenia in this population. So the vast majority of these duplications, about 60%, were de novo, so spontaneous mutations. But 40% of these duplications are inherited. And we know this because we could take advantage of a technique to examine the methylation status of any duplication that we detected in our genome screens. So previously, in this study, we had trios, so we could follow the inheritance of the mutation down through the family tree. But in this study, we didn't have the parental DNA, but what we could use was this high-resolution melt curve analysis to examine the methylation status of any duplication that we detected. So we didn't need parental DNA, but we could take advantage of the increased methylation that occurs on the maternal chromosome, and the lack and the decreased methylation that occurs on the paternal duplication, to understand where these duplications have been derived from. And this, of course, also meant we could follow the inheritance of the duplication through families where it was inherited. So, for instance, here we have an individual with no duplication, and this was the unaffected father. We had the affected individual who had inherited it from their mother and had a maternal duplication. And we had the mother who carried the mutation, but hers was derived from her father. It was a paternal duplication, and so she was unaffected. And you can see this on these family trees of the individuals with schizophrenia. And what you can see in some examples is the way that this mutation can skip generations. So, for instance, in this trio here, we have a father who has no mutation and is unaffected. We also have a mother who does carry the duplication but is unaffected. And that's because our analysis tells us that her mutation that she has, her duplication, is paternally derived. Of course, then she passes this mutation on to her offspring, and it's damaging because of the status of the methylation has changed during her gametogenesis, and she has passed it on with a maternal status. This can also be applied, this technique can also be applied to de novo mutations, enabling us to determine the parent of origin of any mutation that is detected. Finally, I want to just finish on talking about multi-locus imprinting disturbances. And this is a new idea and has come about as a consequence of detailed investigation of a number of incidences of these disorders. So there is increasing evidence that imprinting disorders are caused not just by methylation mutations that occur at one individual locus, such as the Beckwith-Wiedemann locus, but that seem to occur at multiple imprinting loci. And these are termed multi-locus imprinting disturbances. And so these multi-locus imprinting disturbances show changes in DNA methylation at several imprinting DMRs. And the causes of this seem to be a multifold. So there could be damaging gene variants that encode the epigenetic machinery that protects the imprinting loci during this early embryogenesis stage. So if you remember, this rapid demethylation of the paternal and the maternal genomes is protected at imprinting loci by these groups of proteins, the SCMC proteins, and also in particular ZFP57. So for instance, a damaging gene variant of ZFP57, which is not as functional as it should be, means that all imprinted loci have reduced protection from this demethylation process. And this can give rise to multi-locus imprinting disorders. And these may present as one disorder. So for instance, an individual with Beckwith-Wiedemann syndrome may be diagnosed with Beckwith-Wiedemann syndrome on the basis of their characteristics, their genome at 11, chromosome 11 may be investigated and indicated that there is an imprinting disturbance, but more detailed investigation may demonstrate that there are multiple imprinting disturbances at this loci. In addition to gene variants that may be damaging, environmental factors may also lead to multi-locus imprinting disorders. So this period of time here, during this rapid change in epigenetic status, is going to be sensitive to environmental input. And one of those environmental inputs is assisted reproductive technologies, particularly assisted reproductive technologies where there is a period of external fertilization or culture, so in vitro fertilization. So in vitro culture may disrupt the normal epigenetic program, and also imprinted DMRs may be particularly vulnerable at this point. And so this was investigated first by looking at the prevalence of Angelman syndrome in babies born through assisted reproductive technologies. And these studies found that there was an increased incidence of Angelman syndrome. But what was really interesting was when they examined what kind of mutations were occurring in those individuals that had developed Angelman syndrome and were derived from assisted reproductive technologies. So if you remember, the main mutations that give rise to Angelman syndrome are a deletion affecting the maternal chromosome between these different breakpoints, a paternal uniparental disomy, and also epimutations, hypomethylation, which on the maternal chromosome, which gives rise to a paternal gene expression-like system. And what they found was that in individuals in AS who had been born via ART, there was no incidence of these genomic mutations at all. All of them occurred as a consequence of epimutation, so hypomethylation. And so the conclusion was that ART may interfere with the epigenetic reprogramming of the cells and give rise to this increased incidence of these imprinting disorders. Now, it's important to recognize that these imprinting disorders are extremely rare, and although there is a significant increase in the incidence, they are still extremely rare even in individuals born through ART. But nevertheless, this shows that there is a slight risk of increased imprinting disorder incidence following assisted reproductive technologies. So finally, just to summarize, we've gone through the idea of genomic imprinting and the cycle of imprinting. I hope you understand that both maternal and paternal genomes are required for mammalian development, and this is a consequence of the existence of these parent of origin specific imprints, differentially methylated regions, and the consequence of expression of imprinted genes from one parental allele only. These imprinted DMRs persist during the development of the somatic cell lineage, so the inherited DMRs are maintained and then persist throughout somatic cell development, but they are erased and reset during gametogenesis, and this has important consequences for the inheritance patterns of imprinting disorders. So disorders linked to imprinted genes can occur via 4 key mutation mechanisms, and we contrasted a number of different syndromes that occur at imprinted loci, but we also outlined that whilst most of these are de novo, there may be some inherited mutations, and the example I gave was duplications at 15q, and this can give rise to the penetrance of the mutation skipping generations as the epigenetic status switches through different generations during gametogenesis. And finally, we discussed the idea of multilocus imprinting disorders, so how reinvestigation of some imprinting disorders reveals that actually rather than a specific mutation at one given imprinting cluster, it may be a broader change in the imprinting status across all imprinting clusters, and there seems to be an increased risk of epimutations following assisted reproductive technologies. Thank you very much.